Today's topic is cancer heterogeneity. So a cancer tumor can be homogeneous or heterogeneous. Homogeneous means that all the cells in the cancer tumor are similar, not the same, but similar. So even if there is a small mutation, the final function is similar. It does not make a big difference. Cancer tumors can also be heterogeneous. Heterogeneous means when the cancer cells in a tumor are also different. That means there are subpopulations of cancer cells inside the same tumor. Now, there are two types of cancer tumor heterogeneity: inter-tumor heterogeneity and intra-tumor heterogeneity. So, inter-tumor heterogeneity is between two different tumors, let's say two different patients. And intra-tumor heterogeneity is within the same tumor, the cancer cells will be different. Now, I am going to talk about intra-tumor heterogeneity, where we are talking about the same tumor. Now, inside the same tumor itself, why will there be so much difference? It's because of three main factors. The first one, genetic. So mutations. I will speak about this in detail in the next video. Due to mutations, the cells can be different. The second one, epigenetic. Now, if two cells have the exact same DNA, exact same genetic material, will they be the same? The answer is no. Because there is something called epigenetics which decides in two different cells which gene is active or upregulated and expressed more, and which gene is inactive, downregulated, silenced, expressed less. So even if two cells have identical DNA, identical genetic material, epigenetics decides which genes are active and which are not. So that is the second level of difference. And the third, obviously, is the tumor microenvironment. So in the same tumor itself, some areas can have more oxygen, more nutrients, more blood, and some areas may not have that. Some areas may be near a nerve, that's why they cannot be cut, but some areas may be at the periphery and they can be cut. So that is what is microenvironmental heterogeneity. And now this heterogeneity is what makes a cancer so hard to treat. Because in treatment, relapse occurs because of cancer tumor heterogeneity. It's one of the main factors why relapse occurs. I spoke about cancer heterogeneity and its three causes: genetic, epigenetic, and microenvironmental. Today I will talk about genetic heterogeneity in detail. So the first reason of genetic heterogeneity is point mutations. So every time a cell divides, it has to copy its DNA. And during this process, errors can occur. For example, deletion of a letter, addition of a letter, substitution of a letter. And with the constant turnover of tumor cells and the large tumor cell populations, some of these mutations will affect genes with known cancer relevance, like tumor suppressor genes. The second reason of genetic heterogeneity is aneuploidy. Aneuploidy is when a chromosome number is either more than 2 or less than 2. If it's more than 2, it's trisomy. If it's less than 2, it's monosomy. And it's not. The third reason of genetic heterogeneity is polyploidy, also can be called WGD, whole genome doubling. And this is also where aneuploidy arises. So in aneuploidy, one chromosome is more in number or less in number. But in polyploidy, the entire set of chromosomes, all the chromosomes are more in number. For example, all the chromosomes, instead of 2, there are 3 copies or 4 copies, tripled or quadrupled. And this is not very stable. So some of these copies disintegrate, and this is where aneuploidy is born. The fourth reason is chromosomal instability, which is an increased rate of genomic mutational errors involving loss, gains, or translocations of large fragments of DNA. Now here what happens? Chromosomal instability allows a much faster rate of genomic diversification. This helps speed up the process of evolution during tumor development and cancer therapies. So cancer cells with the wrong number of chromosomes mutate much more often than with normal number of chromosomes. CIN therefore is a self-amplifying process. If there is very less chromosomal instability, the tumor cannot evolve fast enough to resist therapy. If there is too much chromosomal instability, the tumor itself is too unstable and dies. But if there is intermediate level of chromosomal instability, that is the maximum danger point. It's just enough diversity to escape treatment and enough stability to keep growing. The fifth reason is extra chromosomal DNA. Normal DNA sits inside chromosomes. Extra chromosomal DNA is a small circular loop of DNA that broke free and floats independently inside the cell nucleus. It's like plasmid in a bacteria. It copies one cancer gene hundreds of times. So instead of duplicating one chromosome, extra chromosomal DNA cuts off only the cancer gene that it needs and makes hundreds of copies of just that one gene. It spreads randomly when the cell divides. Normally, chromosomes will split up between two daughter cells, but extra chromosomal DNA can distribute completely randomly. One daughter cell may get 200 copies of an oncogene, another will get only 5. This is instant diversity at every division, no more mutations needed. And therefore, it drives therapy resistance faster than any other mechanism, because copy numbers vary so randomly and rapidly across cells that when a drug kills most of the tumor, the subpopulation with this optimal ecDNA dose survives and takes over. The tumor essentially dials up the right oncogene level under pressure. And the sixth reason is cell fusion. So we don't exactly know why, but in cancer tumors, cells fuse sometimes. And this fusion results in merging their DNA and mixing and matching genomic fragments, also resulting in diversification, which leads to heterogeneity. See you tomorrow. And today I'm supposed to talk about epigenetic heterogeneity, but I realized before that I should explain what epigenetics is first. So, genetics is like the DNA code that is there in every cell, and epigenetics is what decides which part of that code will be active or inactive. So think of it like this. There's a book, and every single word, line, sentence that's written in that book is DNA. And now you take a pencil and you underline certain lines in words and you cross out certain lines in words. That is epigenetics. You read the underlined parts more often and you do not read the crossed out parts at all. So there are a few interesting examples. There are two twins and twins have almost identical DNA, but one of them develops a disease and the other does not. Why does that happen? Because of epigenetics. The environment can influence your genes and decide which gene stays active and which does not. And that is what allows one twin to have a disease and the other twin to not have the disease, even though the underlying genetics is the same, but the epigenetics is different. The second example that I'd like you to think about is, if you take DNA from a hair cell, your saliva, your blood, your liver cell, heart cell, all of these cells will have the same DNA, right? So why do they have a different structure, a different function? Why do they look so different and why do they perform such different roles? That again is because of epigenetics. So in a cell, epigenetics decides which genes are upregulated, that means expressed more and active, and which genes are downregulated, that means expressed less, inactive. Now there are different ways that this happens. One epigenetic mechanism is called DNA methylation. So a methyl group is CH3. A methyl group going and attaching to any letter on the DNA goes and silences it. So basically that particular gene will be silenced. The second epigenetic mechanism is histone modification. So histones are proteins that the DNA wraps around. The DNA can wrap around tightly, making it difficult to read letters, to read genes, or loosely, making it easier to read the genes and express those genes more. So today I'll talk about epigenetic heterogeneity. 1. Epigenetics changes what genes do, not what they are. So the DNA is the same, but different genes are switched on and off. This has a greater impact and is more powerful than genetic mutations, because here the behavior of DNA matters more than what the DNA actually is. Second, some epigenetic changes are permanent, some are temporary. So some changes like silencing a tumor by DNA methylation, previous video, are stable and heritable, but some changes are driven randomly by noise and can disappear with a few cell divisions. So this spectrum makes epigenetic heterogeneity harder to track than genetic mutations. Cancer cells can shapeshift their identity by something called EMT, which is epithelial mesenchymal transition. So epithelial cells which stick together become mesenchymal cells which do not stick together, lose the ability to adhere to other cells. And this allows those mesenchymal cells to invade, to metastasize, to move to different parts of the body and also resist therapy. So through EMT, triggered by something like hypoxia, which is deprivation of oxygen, a stationary cancer cell can transform into a mobile invasive stem cell. Point number 4, DNA methylation directly drives therapy resistance. DNA methylation is a type of epigenetic silencing where a methyl group goes and attaches to a letter on DNA and silences that particular gene. So MGMT is a DNA repair gene. It fixes chemotherapy-induced DNA damage in cancer cells. So when MGMT is active, cancer cell detects drug damage, repairs it, cell survives. But when MGMT promoter is methylated, that gene is silenced. The cell cannot repair drug damage, the cell dies. The problem is, within one glioblastoma tumor, that is a type of brain cancer, the cells had different MGMT methylation status. Some cells had it silenced, some did not. So when chemotherapy is given, cells with MGMT silenced die, but cells with MGMT active survive. And these surviving cells repopulate the entire tumor. Now the recurrent tumor, the relapse that happens, is temozolomide, that is a chemotherapy drug resistant. So it becomes therapy resistant. I spoke about genetic, epigenetic, and today I'll talk about microenvironmental cancer heterogeneity. So what is that? In a tumor, different regions will have a different surrounding or a different environment due to many factors. One, the cancer cells themselves. Just like I said, due to genetic and epigenetic changes, cancer cells themselves are not the same. They have different types, subclones, subpopulations. So cancer cells themselves are different in different regions. 2, immune cells. There are some immune cells which help the cancer and there are some immune cells which fight against the cancer. In different regions, different populations of these may be present. 3rd, blood supply. Different regions of the tumor get a different level of blood supply and hence nutrients. 4th, oxygen. So low oxygen regions are called hypoxic regions and some other regions might receive an adequate amount of oxygen. 5th, extracellular matrix. So this is like a covering which allows the tumor to spread because this can block drugs. 6th, inflammation. Some regions of the cancer cell are highly inflamed, which promotes tumor growth and survival. And all of these different levels of heterogeneity, genetic, epigenetic, microenvironmental, help the tumor grow back, relapse after treatment as well. Because the tumor is so different, you might be killing only a part of the tumor and the other part of the tumor which is alive helps the cancer population regrow and come back. I'll talk about how heterogeneity results in therapy resistance and relapse in another video, so keep watching. Bye. Talk about how cancer heterogeneity leads to therapy resistance and relapse. So heterogeneity doesn't just cause resistance, it guarantees it. How? 1. Resistant cells already exist before you give any drug. Since the tumor is not uniform, it has many different subclones. Some of these already carry resistant mutations before treatment even starts. These are present in very low numbers, but they're there. So the drug doesn't create resistance, it simply kills those cells which are sensitive to the drug and leaves the resistant ones behind. The drug selected the winner, it did not create it. 2. Treatment itself is a selection pressure. It acts like mutation. If there's a tall giraffe and a short giraffe, the tall giraffe ends up evolving and surviving because it was stronger, it could eat leaves from the tall trees. When therapy kills majority of the cells, it acts like natural selection. The rare subclones that survive are now the fittest. They repopulate the tumor,
about what are the
and
ways
every
that
round of
cancer
treatment
heterogeneity
makes
can
the
be checked.
surviving population
1.
more
Multi-region
resistant
biopsy.
and harder
So
to kill.
taking biopsies from different
3.
parts of the
The
same
recurrent
tumor, not
tumor
just
is
one
not
area,
the same
because
original
different
tumor.
regions can contain
In
different
43%
mutations
of
and even different
glioblastoma
molecular
cases,
subtypes.
at least half of the mutations
2.
in the initial
Longitudinal
tumor
biopsy.
were absent.
Tumors
at
change over time
recurrence.
during treatment and
4.
relapse.
Drug
Repeated
tolerant
sampling helps
persister cells
track tumor
generate
evolution. Like I
resistance
mentioned,
from the
after
start.
treatment,
So let's say
different population of cancer cells survive.
that
So
were
checking those
resistant to
different cells
treatment
is
are not
necessary.
present.
A mutation may appear,
Still,
disappear,
there is a
reappear
population
during
of
therapy.
drug tolerant cells
3.
that
Liquid
survive the
biopsy
initial
and
therapy
circulating
by
tumor DNA.
activating
Tumors,
alternate
cancer cells
metabolic
release tiny
and
DNA
epigenetic
fragments
survival
called ctDNA
programs.
into the blood.
These
And a blood
cells
test
can then
detect
get
these
new resistance
biomarkers
mutations
and
over
changes
time,
and mutations
generating some new sort of resistance that did
ctDNA
not
is
even exist
circulating.
before the
It can
treatment.
represent multiple
5.
sites of the tumor
Different parts
at
of the
once,
tumor
unlike a
respond
single
to
biopsy.
therapy differently.
But
Since
the
different
limitation
regions
here
of
is
the
not
tumor
all
will have a
tumors
different
shed
level of
enough
oxygen,
DNA into the
nutrients,
blood
supply.
so detecting
They
such a
respond to
small
treatment
amount of
also
DNA is
differently.
difficult.
6.
4. Single cell
Monotherapy
sequencing.
always fails
Instead
in
of
heterogeneous
sequencing
tumors.
all the cells and averaging results, sequencing
Different
at a
tumor
single
cells in the
cellular
same tumor
level
have different
reveals
driver
hidden tumor
mutations. That
subpopulations
means main
missed by
mutations.
bulk sequencing.
Monotherapy means targeting
5.
only one
Spatial
driver mutation
single
at
cell
a time.
profiling, like
so if you
cytoph,
target
using
only
cytoph.
one driver
This
mutation,
measures many
the
proteins
other cells
in
with
individual
other driver
cells
mutations
while
survive
preserving
and
the
grow.
tissue
So
structure.
monotherapy
This shows
never
that
works
different cell
in
populations are
heterogeneous cancers.
located inside the tumor.
7. The immune system
Circulating
also
tumor DNA,
gets
ctDNA,
outvoted by
but
intratumor
for
heterogeneity.
brain tumors.
So
And
the
here
immune
we take
system
it from
basically
the
requires
cerebrospinal
a
fluid,
wanted
CSF.
poster
So
of
brain tumors
any
will release
pathogen or
some
anything
DNA in
that
the
it's
blood,
supposed to kill.
and if we take ctDNA
What
from
if
the
that wanted
cerebrospinal
poster
fluid,
only
this can be
depicts 50%
measured
of
in
the
a
criminals
much
of the
better way.
target?
The only limitation here
So
is
how does
collecting
the immune system
cerebrospinal fluid
recognize
is
the other
invasive,
50%?
painful.
And if it doesn't
7.
recognize, how will it
Multi-region
kill
whole
the other
genome or
50%?
exome sequencing. Once again, instead of looking at one biopsy, one part of the tumor, you check the DNA sequence in multiple parts of the tumor. So 8. Autopsy sampling. So autopsy sampling will allow you to see many tumor sites after that to study full body heterogeneity. Reveals major differences between metastatic sites, and it will give a complete tumor map. But of course, this is useful for research, not treatment. 9. DNA barcoding and clonal tracking. Each cancer cell is given a genetic label that is unique. Researchers track which clones survive treatment.